We're going to continue on with absorption mechanisms in the small intestine. Last time we talked about the absorption process without specifics. Now let's look at how this occurs. This is plate 79, and focus first at the top in reviewing transport mechanisms. That was like one of our first lectures, and we're going to come back and we're going to look at them again. The simplest one is by diffusion, based on a concentration gradient. That is relatively nonspecific. Consider that the GI tract has been specialized to pick up nutrients. If you just go up and you pick up anything that comes into your stomach, you could run into some significant problems, things like bacteria or viruses, toxins, etc. So, why not, when you design the system, make the system recognize specific compounds of interest, nutrients that you need, whether it be vitamins, proteins, glucose, or whatnot. The way you do that is by embedding specific transporter molecules. Those transport molecules, proteins, transmembrane proteins, can be active transporters. They may require energy. They can do co-transport. Co-transport means you have to have both products present in order for this to work. And then you can have facilitated, which helps recognize specific compounds and acts as a channel or a port. So then look here at this diagram. These are cartoons of the intestinal cell. So this is an intestinal cell. So this is a portion of the villi. You see there are microvilli, these increased surface areas. And on these microvilli, you see the presence of these compounds. Transporter molecule. Here's this is a co-transport molecule down here. And do we have is there an example of an active transport? No, not on this one. Oh, yeah, right here. Okay, but notice that this transporter is not on the wall of the microvilli. It's on towards the basilar wall, or the wall that faces the capillaries and the lymphatics. This is the capillary. This is the lymphatic. So let's kind of start and look at some of the ways nutrients and compounds are picked up. The first one is iron. We haven't talked much about that iron, other than it's an important component of heme. It is a requirement for the production of hemoglobin. Inadequate iron ingestion or inadequate transfer of iron into your body will result in decreased number of red cells. It will result in an anemia. In our domestic animals, pigs are a classic example of that. That is, pigs that tend to be raised in confinement or on concrete. They don't have access to dirt. They are invariably iron deficient and are required to be given iron shots. So when you see little children, you see little animals out there eating dirt, think of it not as being so dirty, but they're getting iron into their gut. That gut then moves down into the intestine, and you will see the presence of a simple diffusion. But because of the importance of iron, there is a molecule called transferrin that picks up this iron. So what it does is it acts to create a diffusional gradient. It pulls the iron out of solution, binds it to this protein. That means you now still have a gradient for iron to diffuse. there are stores called ferritin inside the intestinal cells. This provides a capacity, a storage capacity for delivery of iron during those times that animals are iron deficient. In fact, in a properly managed environment, you seldom see iron deficient anemias anymore. As that iron is required for hemoglobin synthesis, it is then diffuses into the blood. The next example is facilitated transport for calcium. The importance of calcium ion over and over again. So we've put specific transporter molecules into the microvilli that recognize calcium and move it into the cell. Inside the cell, it gets trapped by a transporter cell or transporter molecule that is involved then, again, of helping bring the calcium in by removing it from its concentration, and then by lowering the concentration in the cell. And then it transports it to the basilar side where it releases the calcium. It's in a high concentration, so simple diffusion into the blood. Here is an example of co-transport, sodium and glucose molecules. Now, if you had thought about this before Mr. Gatorade, you could have made a fortune. What co-transport means is that you need both sodium and glucose present for this to work. These are osmotically active particles. As these particles move in, water follows. Sodium, largest concentration in the extracellular fluid, high demand for sodium in your diet. You ingest a lot of sodium. You need carbohydrates. The co-transport of sodium and glucose by this process results in a huge reabsorption of water. So you've been out exercising, you have lost sodium, you've lost fluids, you want to rehydrate. You should rehydrate not only with fluid, but with salts. And then along comes Mr. Gatorade, who recognizes that it is a co-transport mechanism, that salt will be reabsorbed more effectively if I provide a starch or glucose moiety. So they mix up, and you read the labels on Gatorade. You'll see that there's sodium and also some carbohydrates, all with the principle of I want to get it into the gut and I want to reabsorb it. So when we rehydrate our animals, we like to put in electrolytes, we like to put those salts in, we like to put the fluids in. We also like some kind of energy store. Amino acids, very important, have specific receptors for amino acids to bring those across. Here's another fructose. You can see fructose, another sugar that's being involved in co-transport. They're all being dumped into the blood. Potassium, moving on a concentration gradient. Sodium, here's this active transport of sodium. One thing that's going to help drive this co-transport is if you decrease the sodium inside the cell. How do you do that? You pump it actively out into the blood. That decreases that sodium ion. You now have a great gradient for this co-transport. But this is it's not active, it's not going against a gradient. Example of a vitamin, water-soluble B vitamin. It gets transported by a non-active process, but requires the presence of what's referred to as an intrinsic factor. Intrinsic factors are produced by walls of the stomach. So here's a nice relationship between the stomach and intestines, where in order to get intestinal absorption of a compound, you must have a product produced by the stomach. The stomach produces the intrinsic factor binds to this B vitamin, and then the B vitamin is brought into, enveloped by a process of endocytosis. I'm starting to lose, I'm disappearing. I'm back. So this endocytosis and the movement of this vesicle to the wall, where there is then an exocytosis. So think of all the mechanisms of transport, all of which we've kind of talked about, all taking place in the walls of the gut. Other vitamins, other water-soluble vitamins, can get transported simply by diffusion or by facilitated transport. What about fats? We talked about the lacteals. This is the lymphatic or lacteal. And there are the lipids. But the lipids have to be in a modified form. You've broken them down, but they tend to coalesce, they tend to clump, and having fats of clumps of fat circulating through your blood, bad thing. So how is this processed? We're digesting in the stomach. We've got, can you take me out of the picture for a second? Right at the bottom. So here are our lipids. Lipids are being acted upon by bile salts, the lipases, and are broken down into fatty acids. It is these fatty acids and the glycerol then that are transported into the cells by simple diffusion. These fats are lipid soluble, a lot of lipid in this membrane, so they move readily through the cells. Now, some fats will move, some of the small fatty acids will move right in and move into the blood simply because of this lipid solubility. The majority of lipids, however, have to be encapsulated. They have to be protected with a protein coat. A protein coat, back my arrow. Protein coat that surrounds the lipids. This is now called a chylomicron. Micron because they're very small, and chylo having to do with this lipid component. So chylomicrons are very small because they're coated in a protein. They're water soluble, but they are picked up by the lacteals, not by the blood. So here's some free fatty acids, a small component that are being picked up by the blood. The majority of lipids are transmitted by these chylomicrons. Chylomicrons too big to go into the vascular channel. They can only enter into the lacteals. Lacteals, okay, you bring back. Lacteals dump into the lymphatic ducts. All the lymphatic ducts dump into the thoracic duct. And the thoracic duct takes all the lymph and drops it into the vena cava, drops it into the venous blood supply right before the heart. In that lipid, or in that lymph, you have fluid, you have vascular fluid, interstitial fluid. It's being picked up in all of the tissues. You have some proteins, small concentrations. Now, what we've added to the mix are chylomicrons, lipids. Dog hit by car, comes in, having trouble breathing, thorax moving like this. You draw a sample off, and the sample of thoracic fluid, instead of being clear, is milky. Look at it under a microscope, and you see that instead of having nice, clear plasma, the plasma is filled with these chylomicrons. So what's happened? The trauma has torn the thoracic duct, and instead of emptying into the vena cava now, all of the lymphatics are emptying into the thorax. That includes proteins, fluid, and the chylomicrons. That condition is referred to as a chylothorax. So the chylo referring to those chylomicrons. Functions of the large intestine. What time are we at? 16? Well, there you go. Thank you. Now we are using the human textbook, so some of the variations in the intestines, very intriguing, very supportive of and underlying the evolution of digestion in different animals. But by and large, functions are still the same throughout these different tissues. We're now at the large intestine. This has the cecum feeding in, and in some individuals the cecum is extremely enlarged. The cecum then drops into the colon, and these terms are germane for human: the ascending, transverse, descending, sigmoid colon. Those terms tend to be more human-specific. When we look at our domestic animals, we have different shapes, different orientations of the colon. So it's not important that you know these specific names, other than you have the cecum, which is right at the junction of the ileum, and then you have the colon. And the colon then exits by the anus, and the terminal portion of the colon is called the rectum. So what's occurring in the large intestine? Compaction of the feces. You've got intestinal fluid that's moving through there. This soup of digested material that you're absorbing the nutrients from. Well, when it gets to the colon now, the idea is to reabsorb as much of that water as possible. So the reabsorption of sodium and water in high amounts. So there's a tremendous amount of absorption, both of sodium and of water. There's still peristalsis. You still have moving and segmentation, forming of the little fecal balls and the passing of the fecal balls. Dietary fiber and bacteria aid in colon functions. So an appropriately functioning bacterial flora serves an important role in maintaining homeostasis or normality of the colon. That means alterations to those bacteria can produce enteric problems. So if you've generated this nice little bacterial population that's working really well, and you take a drug, you take an antibiotic, some of those antibiotics, and one of the great ones is erythromycin in horses. Good antibiotic, broad spectrum. You treat it. You treat the horses with erythromycin, and one of the problems is that it kills off the specific populations of bacteria in the colon. The result is an inability to appropriately absorb water and sodium. That means water and sodium that aren't being absorbed are exiting the animal, and you now have diarrhea. So let's look at some of the disorders. So plate 81. Hypermotility of the GI tract. Right, you've been there. Nervous about an exam, nervous about a job interview. So there, in fact, is the vagus nerve can play an important role. Am I on 81 or 80? Okay, it should be on 81. I'm just not at the top. All right, so here's the fear, anxiety operating through the vagus nerve. You can get diarrhea due to inflammation of the gut, a gastritis, an enteritis. If you are inappropriately absorbing material, you're going to carry it out through the rectum. If you can't absorb glucose, if you can't absorb amino acids, you will see diarrhea. Consider pancreatitis. No pancreatic enzymes. No digestion. So you now have particles which are undigested that are going to carry with it water. And those particles will compete with that water along with your gut. Your gut wants to absorb sodium and wants to bring in water, but if there are undigested particles that are too big, they will compete, and you can get diarrhea through undigested food. Certain diarrheas can be caused by an enzyme deficiency, a problem in the digestive process. So let's say you have you're missing one specific enzyme. And we'll say that enzyme is lactase. Lactase, it's an enzyme that is involved in the breakdown of a carbohydrate moiety called lactose. Some of you may recognize lactose as being a milk sugar. So in fact, cow milk has large concentrations of lactose in it. You consume lactose. Without lactase present, lactose is not broken down. It then serves as undigested food particle. An undigested food particle that is in competition with water. As a consequence, lactose and the water associated with it exits. Lactose intolerance. A lot of you know people that are lactose intolerant. That is, they lack the presence of lactase. Consider the dairy industry. Very big, very powerful. They want you all to drink milk. My heritage and my background is Germanic and Scandinavian. We have plenty of lactase. Interestingly, particularly African origin individuals tend not to have lactase. So one of the great problems, with a certain physiological irony to it, was had individuals starving in Biafra, and so what was being shipped was dehydrated milk, powdered milk. Great, high protein, high energy, lots of lactose. So they would these individuals that were in a nutrient-starved state then were being fed lactose. And while there were lots of nutrients that could be absorbed, the presence of lactose, undigested, tended to keep water with it, and these individuals would oftentimes present with diarrhea. Not a good thing when you're compromised. So we see lactose intolerance, a genetic proclivity to it. We can identify these individuals. Clinical signs. You have cheese, you have ice cream, you have milk, and you develop diarrhea. One of the ways to address that is by taking a lactase. So you can find lactase pills, or you can find milks that have been treated to remove the lactose. So anytime you don't digest something, you are going to carry water and fluid out. The other example that they don't give in here that I would like to introduce is the presence of specific bacteria which produce toxins. These are what are called enterotoxogenic bacteria. These bacteria produce toxins, so they are genesis producing toxin in the gut. These toxins actually will act to increase secretion. Now, we talked about the colon being very important in absorption. That's where most of the absorption occurs. It also plays a role in secretion. And if these cells are challenged by this toxin, instead of absorbing water, they will produce a secretion of salts and water and the attendant diarrhea. In some cases, this is what can actually be referred to as explosive diarrhea. These animals have huge amounts of very loose, watery diarrhea. That can produce severe fluid and electrolyte losses if left untreated. Vomiting. When we talk about animals presenting for clinical problems, I think you have to put number one on the list as enteric or GI problems, particularly in our companion animals where you've got Berber carpet at home, you've got a dog that's vomiting or has diarrhea. That is a big issue. You have a vomition center in the medulla. So there are cells which respond to toxins, that respond to emotional distress. A center that can in fact respond to sight or to taste and olfaction. Right? That smells so bad I would just like to vomit. Well, in fact, you have neural control over that. So there is a what is called a vomiting reflex. And you've all been there. Kind of that sick feeling. Glottis closes. You want to shut off access of that acidic vomit to the trachea. Opening the sphincter, the cardiac sphincter. Contraction of the walls of the GI tract. Abdominal muscle. Diaphragm. So all of the muscles, smooth muscle, skeletal muscle, get into this to reverse the flow of material and bring it out through the mouth. Now you think about how protective that is. If I ingest a toxin or a particularly bad product, vomiting serves an incredibly important role in trying to remove that. You're removing it and removing it from the access to absorption. Some animals don't vomit. Ruminants, horses. Are they likely to get into as many toxins? Certainly not bacterial. Carnivores, omnivores, animals that are eating meat, flesh products, there's more likelihood of there being bacteria and toxins in those. Not so much in wheat. So we see the vomiting reflex being very important in our carnivore and omnivores. Ulcers. We now recognize that erosions of the gastric mucosa have as their primary stimulant populations of bacteria. Heliobacter. That's true in humans. That's true in small animals. It's not as true in large animals. But whenever an animal presents with a gastric ulcer, you need to think of a bacterial cause. So you have bacteria that are amplifying themselves, they're multiplying, and they start degrading the mucosal coat. They start removing that mucus covering. The stomach acids then start acting on the walls of the stomach. That erosion of the mucosal surface is called an ulcer. The erosion can be so severe that the animal bleeds out into the stomach. So that's a bleeding ulcer. If the ulcer becomes so pronounced that it pokes a hole through it, that would be a perforating ulcer. Perforating ulcers will kill you. Instead of dumping acid contents into the duodenum, you dump acid contents into the abdominal cavity. Scarring, degradation, and you kill tissue. So that'll kill you. We see that in pigs here on campus where they get stressed and develop gastric ulcerations because of the stress and perforate. Approaches to that: treat the Heliobacter, antibiotics in some instances, decrease acid secretion. So there are a number of pharmacological products on the market that decrease hydrochloric acid secretion. For a while, that was the number before Viagra. That was probably the number one prescription drug, was the compound to reduce hydrochloric acid secretion. What else? Oh, duodenal ulcers. You can have gastric ulcers. You can also have duodenal ulcers. Duodenal ulcers, very painful. Erosion of the intestinal walls. Diagnosis of gastric ulcers. One very, very prominent gastric ulcers, in which you have blood going into the GI tract, will present with very black, tarry feces, because the blood is digested. If you add blood to an acid environment, it deprotonates, it becomes this black, clumpy mess. It then passes through the intestinal tract. We don't have denatured hemoglobin transport mechanism, so it's in the feces, and it gives the feces an incredibly dark, they're black, very sticky because of the protein, a condition we called melena. They also have a horrific odor. If the blood is being produced, and it's not being produced in quantities that you can visibly see, we say that blood is hidden. And the other word we use for that is occult blood. So another test we can run on feces is to measure for the presence of occult blood. If it's present, it indicates an ulceration somewhere up the GI tract. In old animals, if you find the presence of occult blood, you think of the possibility of tumors. Erosions in the wall that have caused some bleeding, small amounts. And a simple diagnostic test will give you an indication of the presence of blood. So we have occult blood. We may have melena from these from the bleeding. The constipation. Cats are great ones to get constipated. Not drinking adequate amounts of water. The animals are hypohydrated, so they tend to absorb large quantities of water out of the large intestine. Results in the production of very dry fecal material. Treat these with oil and water enemas, which, if the cat is fairly sprite and alive, is a bit dangerous. But a lot of these animals will come in very obtunded because they're in hypohydration, and you need to address the issue of appropriate fluid. The last condition that I want to talk about is our friend diarrhea, associated with viral infections. Dogs and parvovirus. What an interesting organism. The parvovirus finds its home in the walls of the intestine, in the mucosal cells. So the parvovirus then goes into these mucosal cells, multiplies, causes destruction of that cell. That cell loses its absorptive capacity. It's no longer absorbing substrates, it's no longer absorbing fluid. So the clinical signs of these animals, diarrhea. But in addition, because there's cell destruction, you have kind of this opening of the capillaries, and you see a bloody diarrhea. Because this is so prolific, it spreads throughout the intestines, you can see not only... Can you bring me back to life for a second, Bo? I'm back. Large expanse of the intestine will be invested with these viral particles, and the diarrhea can actually have drops, not of black tarry feces, but you'll actually see blood in the fecal material. So you have a number of issues associated. You have an erosion of the wall. You now have access to the capillaries, the blood supply to what's in the intestines. That includes bacteria, so you need to look at the potential for secondary bacterial infections. The animal getting bacteria in response to this viral attack. Water loss. But even if the animal recovers, one of the sequelae to parvovirus infections is the replacement of mucosal cells by scar tissue. You kill the cells. They don't regenerate. They are replaced by connective tissue which has no absorptive capacity. So you may see some of these parvo puppies that have recovered. They're doing pretty well. They may be smaller in stature. They may have a stunted growth. Because even though you're feeding them what is perceived adequate amounts of nutrients, they're not able to absorb it. They may also have lifetime diarrhea or loose stools, simply because their absorptive function is compromised. As long as you can keep up with the salt and fluid losses in diarrhea, you have a good chance of dealing with a problem. So yesterday I had a calf that came in off about 100 pounds, flat out in the field, unable to rise, dehydrated. looked around at the tail head. This animal had profuse diarrhea. It had just gotten into that cycle where it had lost too much fluid to keep itself upright to move. Response was to get fluids and electrolytes back into that animal. You can do that oral. So when you get diarrhea, you got the virus, the flu bug, you're trying to keep adequately hydrated. If you're unable to do that orally, then the next step is to go to IV fluids. In fact, we did IV fluids on that animal, and she's doing well today. So that concludes the GI tract.